Okay, hi. Okay, this is part 3 of this unit's lecture, and I had talked about transcription, translation, replication, a little bit about the chromosomes, arrangements of the chromosomes within the bacterial cell, and then tonight I talk further about another type of gene transfer, horizontal gene transfer. In the previous lecture, I had said that there's a vertical gene transfer, which is binary fission, and also horizontal gene transfer. Tonight we're going to go into, or today I'm going to go into the three processes, different processes of horizontal gene transfer. So the name of those processes are transformation, transduction, and conjugation. So if we look at the diagram on top there, this is transformation. And what happens in this situation is that a bacterial cell, the donor cell, dies. You know, they get old, they live, they die. When that bacterial cell dies, the DNA or the chromosome within it starts to fragment and decompose. And it will be released into the environment. Now, what happens is if there is a competent bacterial cell in that environment, another bacterial cell that is competent, it will be able to pick up these fragments of DNA from the donor cell. And those fragments would then be incorporated into that recipient cell's chromosome or DNA. So this is one way in which DNA from one bacterial cell, which has died, is picked up and it is transferred into another cell, the recipient cell. That is one process of horizontal gene transfer. Now, another type of gene transfer is called transduction, which is B, the middle diagram there. And in this case, we have bacterial genes that are picked up by a virus. Okay, so in this case we have a bacteriophage that has infected the donor cell, and as it's in there, of course, it is once it's entered and penetrated and entered the donor's bacterial cell, it will make more of itself and then assemble itself. And as it makes more of itself, it may pick up some of the donor cell's DNA. And once it's assembled, then it will break out of that donor cell, carrying a little bit of the that cell's DNA, and then transferring it to another new recipient cell. So it's the donor bacterial DNA that will be injected into the recipient cell, along with the virus, the bacteriophage's genome. And once that happens, it will become, again, part of the recipient cell's DNA. So what this means is that, especially in transformation transduction, this new piece of DNA or this segment of a gene will become part of the recipient cell's DNA. And as that bacterial cell replicates, it will replicate also the genes of the donor bacterial cells to pass on to further generations. Okay, the last thing is of horizontal gene transfer is the process of conjugation. So in this case, you'll have one bacterial cell that has a plasmid. Usually it's the F plasmid, which, remember, carries fertility genes. And what that means is that plasmid, when that gene is expressed, will be able to build a conjugation pilus. Okay, a conjugation pilus. So you've got one bacterial cell that has a plasmid that contains the fertility genes. It will, when expressed, it will build a pilus or a tube that will connect to a recipient bacterial cell. Once that occurs, then the plasmid from the donor cell, basically one part of that DNA from the plasmid will be transferred into the recipient cell. And once it's in the recipient cell, it will replicate so that you have a full plasmid. And the same thing with the donor cell. It's given one part of its plasmid to the recipient cell. It will also replicate, so it's a complete plasmid. Okay. So another thing with conjugation too is that not only will the entire plasmid be sent, or be given to a recipient cell, you also may have some of those genes, those transposons, those jumping genes. Remember I had talked about that beforehand, before conjugation took place, a segment of the DNA from the chromosome itself would have jumped into that plasmid, and that gene from the chromosome will then be part of the plasmid, and that will also be transferred into a recipient cell. So basically the general process of conjugation is that it's the transfer of a plasmid, not the whole chromosome, but the plasmid from one cell to the other. The interesting thing is these plasmids are also can be fertility plasmids. They may be plasmids that code for antibiotic resistance or for toxins. So I'm going to talk a little bit about the history of the first process, transformation. That was discovered in about 1944 by a scientist by the name of Frederick Griffith. And what he did is he had taken 2 strains of Streptococcus pneumoniae. One strain was non-toxic, and it did not have a capsule. It was not pathogenic, and they call it R strain because the colonies themselves were rough looking. But again, the R strain was without a capsule, and it was not pathogenic. Then there was another strain of Streptococcus pneumoniae that had the capsule, and the colonies were kind of smooth looking, so they called it S strain. What they did know is that with the S strain, it was pathogenic, that the organism that was infected with it would die. So they did a little test. When they had the ones with no capsule were injected into a mouse or into an organism, the mouse lived. So again, it was non-pathogenic. Those that had the capsule, the S strain, it was proven that if they were injected with that strain of Streptococcus, the organism would die. So they set up this experiment. They ran the control with the non-capsulated Streptococcus to show, yes, it was not toxic, it did not cause death. What they did then, knowing that those that were capsulated, the Streptococcus that were capsulated, they killed them. They killed the bacteria before injecting it into the mouse. And what happened is the mouse lived. He did not die as expected. So in experiment 2, they combined both the rough strain and the heat-killed smooth strained bacteria, and then injected that into the mouse, and the mouse died. They took the bacteria from that dead mouse and injected it into another mouse just to confirm that what they saw was true, and in fact the mouse did die. So what happened? They figured and correctly figured that non-toxic R strain had picked up some of the genes from the heat-killed smooth strain. And that was exactly what happens with transformation. Okay, so this is what happens in the real world out there, that the bacterial cell will die in transformation. And again, another bacterial cell, which is competent. Now, by the word competent, I mean that it is capable of picking up those segments of DNA. Not all bacteria are competent, are not equipped to pick up remnants of DNA from the environment. But if they are, then that DNA from the original cell will become part of their genome. We've used this knowledge to apply to medical technology. And in this case, they're in the right-hand side, they're showing you the lab applications of this transformation. And they have a human cell. Basically, they have the gene for the human growth hormone. There are people that do not have sufficient amounts of human growth hormones, and that is a whole area of disease. But if they get from a human cell, they will take a gene, a normal gene for human growth hormone, and they are able then to incorporate this gene into a bacterial phage. And then through amplification, they will have human growth hormone produced. So this is a transformation at its best in helping people. So this is a new technology. The second process is transduction. And again, I want you to look at that note that you need a virus, a bacteriophage, in order for this to occur. Now, in transduction, or even in the infection cycles of bacteriophages, we usually talk about the lytic cycle, where the virus will infect its genome into a bacterial cell. And of course, then you will get the process of biosynthesis, assembly, and release. And then at the end of this, the bacterial cell itself is killed upon release. In the lysogenic cycle, this is another cycle in which transduction can occur. In the lysogenic cycle, what happens is the virus will infect its genes into the bacterial cell. It then the genes of the virus will become incorporated in the genome of the bacterial cell, and this is now known as a prophage. And it doesn't immediately make new parts or release new viruses. That genome will basically just sit on the bacterial chromosome for a while. At some point, and they're not quite sure what targets the startup of its assembly and release, but at some point, the virus's genome will then be released or become part of, or get out of the chromosome, and will start going through the process of biosynthesis and assembly, and then finally making new viruses. And what happens is when the virus's genome leaves the bacterial genome, it may take a little piece of that bacterial cell's genome with it. So when that virus has assembled into new viruses, not only does it have its genome, it will have a piece of the bacterial genome in there with it. And what it does, it will go on to infect, like I said, new viruses, not only with injecting its genome, but a little bit of the bacterial cell's genome. So again, this will as the bacterial cell acquires this new genome, it will start replicating, and then you will get scores of bacteria carrying a little piece of this genome from a previous bacterial cell. Okay, finally, the last process, conjugation. Again, this is the transfer of genes on plasmids through a conjugation pellis. So I explained a little bit about that in the earlier slide. In this case, A is talking about the general process of conjugation. You've got a plasmid, the pili will form, and then what happens is one strand of the F plasmid is transferred into the new recipient cell, and then the donor cell will make a complementary strand of the restored plasmid, and the recipient cell will also make the complementary strand of the plasmid. So you end up with two bacterial cells with two F plasmids. Now there is another way in which conjugation also occurs, and this is when the plasmid is inserted into the bacterial chromosome. So in some cases you do have those plasmids that kind of jump back into a chromosome. And what happens when you get conjugation is you'll get the conjugation tube or pili formed. But then that plasmid that is piece of the chromosome now will start to transfer into the recipient cell. So not only do you get some of the plasmid, you may, in this case, get part of the donor's DNA. But this isn't as common as situation A when we think of conjugation. So finally, we're talking about diversity, and this is just a nice little diagram showing you what causes diversity and what does diversity lead to. So we get things such as mutations, which we really haven't talked about. It is in your textbook, but just natural mutations where you have a misalignment of the nucleotide bases, or an elimination of the complementary bases, and what you end up is with a natural mutation. That provides diversity within the environment within a population of bacteria. You also have horizontal gene transfer, which I've just talked about, transferring genes to neighboring bacteria. This results in diversity. Also, if we look at the orange block, first orange block here, I didn't talk too much about this, but this is another way in which diversity occurs, is when you have genome reduction. Now, with bacteria, what happens when you take a piece of the genome and basically it just stops expressing a gene? That happens when you get in bacteria that basically are intracellular bacterium. So in other words, they've infected a bacteria. They live within that bacterial cell. And that's all they do. They're pathogenic, but they live within that bacterial cell. They won't need the genes for some of the metabolic pathways, because now they can use the enzymes for those pathways from the cell. So just to be more efficient, because the bacteria are very efficient, they will drop or stop producing those, expressing those genes, no longer needing those genes. So diversity can be due to genome reduction. Again, and genome acquisition through horizontal gene transfer, and finally mutations. Okay, and what does this lead to? Diversity. If it is favorable, the diversity is favorable by natural selection, that then in the long run will lead to evolution of new bacterial species. And one of the hot topics today are the arising of bacteria that are resistant to all antibiotics. Again, since the discovery or invention of synthetic and natural antibiotics, we have gotten to this point where we've got bacterial species that have evolved that have become resistant to all of the known bacteria that we know today. So this leads into this movie, Hunting the Nightmare Bacteria. These again are about bacteria that have become either a problem to eradicate in disease, which it focuses on three situations, or it's about bacteria that have become resistant to all antibiotics. So we're going to see these three cases, and we're going to see how that is dealt with. The two individuals, it's Addie is the girl, I think she's about 12, and then there's David, who must be about 19 or 20. And then finally it's about a hospital situation that has become infected with the antibiotic resistant bacteria. So again, please give this a watch. There'll be a few questions on the exam about this. All right, see you soon, and have a good weekend if I don't see you before next week.